Good morning and afternoon, ladies and gentlemen, and thank you for joining us. I'm Felicia Hawk, your moderator for today's webinar. If you have any questions during the presentation, please submit them using your control panel, and we will get to them at the end. I am pleased to introduce Heather Wetzel, who will lead the webinar titled Achieve High Diagnostic Yield for Clinically Relevant Genes, How to Ace Exome Sequencing, to discuss our recent publication in Genome Medicine. Heather is a genetic counselor and member of our field applications team who brings years of clinical experience from prestigious institutions such as UCSF, Stanford, and Northwestern. Please welcome Heather Wetzel.
Thanks for the introduction, Felicia, and thanks everyone for joining us on Friday. So as Felicia mentioned, the primary purpose of today's presentation is to review some highlights from our recent publication in Genome Medicine, and also to give a little bit of background about what we do at Personalis and our ACE platform, which we'll talk about quite a bit in this presentation. So as you can see from this outline, first what we'll talk about, again, as Personalis and who we are as a company and some of our products at Personalis. And then we'll go through some of the highlights of the recent publication in Genome Medicine. And then we'll also sort of switch gears a little bit after that and talk about how the publication and what we do at Personalis really plays out in the clinical setting by talking about our ACE Clinical Exome test and the diagnostic yield associated with that test, and also highlight a couple of case studies that talks about performance of our ACE Clinical Exome. And of course, at the end, we'll save some time for questions and discussion. So for those of you who might not be familiar with Personalis, we're a private company that was started in late 2011. The company was founded by John West, who's our CEO, as well as 4 Stanford professors, and those are Atul Butte, who's now at UCSF, Russ Altman, Ewan Ashley, and Mike Snyder. We've grown quite a bit since the time that we were founded in late 2011 and have employees all across the world. Most of our employees are housed in Menlo Park, which is where Personalis is based. But we also have employees who work remotely in California, also in the UK, and even in Australia. Noted here that we have a CLIA/CAP lab, and primarily we focus on cancer and Mendelian syndromes. And so as we'll discuss in more detail, our entire library of services at Personalis is built on something we call the ACE platform. ACE stands for Accuracy and Content Enhanced. And that platform and every step of that platform is really the basis of everything we do at Personalis. We have products for researchers and engaging in large-scale sequencing and annotation studies, as well as smaller studies with Mendelian genetics. We also are rapidly advancing our presence in the cancer space with cancer studies for biomarker discovery, clinical trials. And today, last but certainly not least, we'll be focusing more on our clinical grade testing via exome sequencing for our ACE Clinical Exome, as well as the newer partnership that we formed earlier this year with Athena, performing Athena's Neurome, which is a whole exome sequencing test that's geared towards neurological phenotypes. And so what you see on this slide here are the various steps that are involved in next generation sequencing. And so, you know, one part to point out early on when we developed the ACE platform is we recognized that there were a number of problems at really each step along the way in the workflow of next generation sequencing. And our goal with ACE was not just to accept those problems and create a product that's similar to what's available in the public, but really to improve on each step of this process and to make an augmented or a much better product than what might be available. And so that's really the basis of what we do at Personalis. Our recent publication in Genome Medicine that we're going to talk about really focuses on sequencing the most. So that's what we'll talk about for most of this presentation. So what we'll do next is review Personalis' recently published Genome Medicine article. One part I want to point out in this slide here, which is the title page for our article, is that, of course, most of the authors that you see listed here are prior or current employees of Personalis. There's a couple of other names, though, that I'd like to point out, and those are Justin Zook and Mark Salet, and these are individuals who are employees of the National Institute of Standards and Technology, or NIST. And we've of course worked with them and collaborated heavily for this particular publication, but also for other work. A little bit of background. Again, I was saying that exome sequencing is commonly used more and more in the clinical evaluation of genetic disease. However, the variation of coverage and the sensitivity over these medically relevant parts of the genome is still relatively poorly understood. There's a number of sequencing-based assays out there that will provide coverage that's really inadequate for clinical assessment. And so, as an example, there may be clinical exome sequencing tests that would say cover 90% of coding content on average, but by providing 90% on average, there's going to be areas with wide gaps of coverage, and these are really important areas, particularly in the clinical setting, because if there's no coverage in an area where a variant resides, then obviously that's going to lead to a misdiagnosis for that patient or family. And so that's exactly what we wanted to look at in this study. We wanted to really focus in on coverage and accuracy that's obtained with these currently available exome capture technologies, and we also wanted to look at whole genome sequencing for clinical applications. Our second objective was to evaluate the ability of these assays to reach a clinical standard of coverage of these key medically important genes of the exome and medically important regions of the genome. We do this because we wanted to provide a clear set of analytical performance measures to aid you, the clinician, in really evaluating and making decisions about choosing laboratories for genome scale testing approaches for these clinical applications. And so to do this, we used sequence data that were all obtained from the same reference sample, which is NA12878. We used predefined lists of medically relevant protein coding and non-coding sequences. We compared coverage breadth and depth across 4 commercial exome capture platforms to do this. And we also compared our own performance of Personalis' augmented exome strategy, which is ACE. So what you see here are the various platforms for which sequencing data were obtained, and then these are the acronyms that we used in our publication and in some of the figures that we'll show later on. And so just to walk you through what each one of these are, the first, of course, we've talked about before, which is ACE, our Personalis Accuracy and Content Enhanced Clinical Exome. The second one you see here, SSCR, is Agilent SureSelect's Clinical Research Exome, followed by Agilent's SureSelect version 5 with the UTR. NX is Illumina's Nextera. NG is Nimulgen's SeqCap version 3. And then WGS, of course, is Illumina's 31X whole genome sequencing. One part to point out here is that numbers 2 through 5 here, these are the exome sequencing capture kits that are most commonly used by other clinical service laboratories as their exome sequencing capture kit. And so a little bit more information about what ACE is specifically. Of course, we've already mentioned that ACE stands for Accuracy and Content Enhanced. The purpose of ACE is really, as I've mentioned, to address coverage and accuracy issues that are found in conventional whole exome sequencing platforms and also found in whole genome sequencing. With ACE, we include standard whole exome sequencing, but then we're enhancing coverage of medically relevant genes. And for ACE, we're specifically targeting in that enhancement approximately 8,000 genes of either known medical relevance or genes that may in the future become medically relevant based on some early animal or case studies, for example. Our goal with ACE is to achieve consistent and uniform coverage across genes of known medical relevance. And this is a term that we coined gene finishing, and we'll talk about exactly what gene finishing is in just a few moments. One part to point out with ACE is that our coverage isn't limited to the exome as other exome sequencing providers. We're also targeting key intergenic and intronic regions of known medical relevance in our ACE design. And so what you end up with ACE is not just exome sequencing, but exome sequencing plus targeting other non-exonic regions of relevance. So what I'm showing you here in this cartoon is really how our ACE or additional capture really works. And so just to sort of orient you to what's going on in this cartoon, and this is just using any gene as an example, what you see here in the yellow all the way across here are your exons in a given gene. What you see in blue are what parts of those exons might be covered by a standard exome sequencing capture kit. And so what you would see here is that, of course, there's some exons that are covered completely, some exons that aren't quite fully covered, and then, of course, there's going to be some exons that have no coverage, and these could be sometimes first exons, for example, due to high GC content. And so what we do with ACE is we're actually building on top of what's already present with the blue with our augmentation. And so what you see in green is our enhancement that's provided by ACE to really fill in and complete these areas where there's poor coverage. And so when you put those two together, what you end up seeing is a much more complete or finished sequence across those coding regions or across these exons. And so then the coverage becomes more complete, even if exons that aren't covered at all are now covered by providing that supplementation. As I pointed out earlier in the prior slide, another part to draw your attention to are these triangles that you see here. And these are areas that, of course, aren't covered at all by a standard exome, so there's no blue coverage here. But these are those biomedically important regions that are outside of the exon. So these are also specifically targeted in our ACE design. What you see listed here are some of the technical specifications from the Genome Medicine publication. And not to go into all of them in detail, but one of the points I really want to stress here is that the data for the purposes of this study were downsampled two different ways. One to 100x average coverage and another to 12G or 12 gigabases of output between data sets. And we did this because we wanted to be able to normalize the data as much as possible across that target set. Of course, all of these platforms really differ in their overall sequencing footprint, and so by downsampling, we were able to really try to take that part out of the equation. For the figures that we're going to talk about in this paper or in this presentation, we're looking at data that were downsampled to 100x coverage. We did also share the results from the downsampling to 12G in a supplemental publication related to this, and the data were very similar. So as I mentioned, the study focus is really clinical in nature, and so we wanted to really assess performance over clinically interpretable genomic regions, and we wanted to look at those performance characteristics for making correct diagnoses as well as minimizing the risk of misdiagnoses or false negatives in a clinical setting. And so we looked at a few different things here. One is something called gene finishing of the medically interpretable genes, and we'll talk about what both of those concepts mean in a couple of slides. We also looked at coverage of difficult to sequence, but also biomedically important regions of the genome, and so again, particularly areas of, say, high GC content would be something that would fit here. And then, of course, we wanted to look at coverage of the 56 genes and known disease-associated loci within those genes that are recommended by ACMG for reporting of secondary findings. So let's move on now and talk a little bit about this gene finishing concept that we've mentioned a couple of times up to this point. Just a little bit of background. Again, gene finishing isn't really a concept that's commonly used in clinical diagnostics, although it probably should be as compared to average coverage, for example. Basically, with next-gen sequencing, we're looking at many sequences that are typically around 100 to 125 base pairs in length, and then those sequences are stacked up in a line to a reference genome, and so that's sort of what we're depicting here in this picture. Some of these areas are going to be very well covered, and then of course some areas aren't going to be well covered. And the problem with that is when you're focusing on this concept called average coverage, of course it's also, you're going to have these areas where there is a great deal of coverage or sometimes too much coverage, but you're also going to have areas where there's not enough coverage. And so, there was a paper that was published a couple of years ago by Maynard et al. That said in the clinical setting, you really need to have at least 20x coverage across every coding base to have high confidence in your data calls or in your small variant calls. So if you have 5x or 10x or 0x, you really don't have the confidence that you need to make variant calls in the clinical setting. And so again, that's why this average coverage isn't truly sufficient, because some areas aren't going to have that level of coverage. In this study, that's why we focus on gene finishing, and this is also what we focus on for everything at Personalis, is how well we finish a gene. And this is looking at having 100% of the gene's exonic bases covered at least 20x. And of course, this probably sounds like a really high or stringent standard, but in the clinical setting, at Personalis, we feel that that's absolutely essential to make sure that we're being as accurate as possible in the variant calls that we're making, particularly in the clinical setting with patients and families. And so to evaluate relative platform performance, we calculated coverage and accuracy statistics for ACE as well as the other platforms we've mentioned within commonly defined medically relevant target regions. And so these are two regions that we have listed here. The first one is the NIST or NIST genome in a bottle high confidence call set. This particular call set is restricted to, as you would imagine, high confidence regions among multiple platforms, aligners, and variant callers. And as a result, regions of the genome where the likelihood of an incorrect genotype is increased, those are actually removed from this call set. Because of that, we also utilize the second call set that contains variants that fall into regions that are difficult to sequence. This is really useful for evaluating these problematic regions that are typically excluded from the prior high confidence call sets. So as I've mentioned a couple times, we looked at gene finishing and call accuracy across what we call the medically interpretable genome, or MIG. And this slide goes into that a little bit more detail in terms of what is the medically interpretable genome. And so specifically, it's a set of 5,419 genes that have mutations in those genes that are associated with disease or disease-related drug response. These are genes that are part of an existing clinical test. They've been documented in the literature as pharmacogenes, or they have a causal association with Mendelian disease, inherited disease, or cancer. As you can see from looking over at the figure on the right, most of these genes are from 3 public sources, and those public sources are OMIM, the Genetic Testing Registry, and HGMD. We did supplement with genes from the Cancer Gene Census, as well as PharmGKB's list of very important pharmacogenes, or VIPs, that have dosing guidelines. One other part to really point out here, just to make sure this is clear, is that prior, and a few slides prior, we talked about supplementing approximately 8,000 genes with ACE. And I just want to be clear here that here we're looking at, for this paper, we're looking at these 5,419 genes not the entire 8,000 genes that ACE is supplementing. For the purposes of this study, we really wanted to make sure that the genes we were looking at were genes that were part of existing clinical data sets. So that way there's no disagreement between whether or not a gene is truly medically important. And so to go over some of the results of the study, we've got a few diagrams that were pulled from the publication to talk about in a little bit more detail. And so the first one that I want to go over with you today is the results looking at coverage efficiency in the medically interpretable genome. So again, looking at that list of 5,419 genes. On the left panel, what we're looking at is coverage efficiency in the coding regions by platform. And on the right panel, we're looking at coverage efficiency in the non-coding regions. And so moving over to the left side, what you see here on the y-axis is the percentage of medically interpretable genes with anywhere between 90% to 100% of the exonic bases covered. Looking at depths on the x-axis, anywhere between 0 and 60x. The dotted line is showing here 20x coverage, which again, remember, is that sort of clinical standard minimum threshold for coverage that we look at in again, the clinical setting. And so what you can see here, looking at this 20x threshold, is that ACE in green has the highest performance of efficiency or coverage efficiency at approximately 99% of the bases covered at 20x, followed by very closely, probably a tie between Nextera and whole genome sequencing, and then as you can see, the rest of the exome sequencing platforms are following underneath that. If you scoot over to the right-hand side of the screen and look just at non-coding regions, obviously that's going to look a little bit different. And again, what we're looking at is the fraction of bases covered on the y-axis here going from 40 to 100%, and depth again looking from 0 to 60x. This time what you see is whole genome sequencing is sort of winning this race and having higher coverage efficiency for non-coding regions, but ACE is very, very quickly behind whole genome sequencing. And again, that's because these are regions that we're specifically looking at or targeting in our ACE design. As you might imagine or would expect, the exome sequencing providers are going to fall much lower down on this list because these are areas that exome sequencing capture kits really aren't focusing on. One exception to that is the one that you see listed here, SS, and this is Agilent SureSelect with UTR. Obviously, there is some non-exonic content as part of their design, but for the most part, what you're really going to see in the non-coding regions in terms of performance is ACE, our exome sequencing at Personalis, and whole genome sequencing. So of course, we also looked at gene finishing in the medically interpretable genome, and we talked about this concept of gene finishing already. What you're looking at here is the relationship on the left side of the percent of finished exons on the Y as the coverage stringency varies and increases on the x-axis. So here we're going from 90% of bases covered at a minimum of 20x to 100% of bases covered at a minimum of 20x. On the right-hand side, we're looking at it a little bit differently, and we're looking at, on the y-axis, the percentage of finished exons with 100% base coverage as the read depth varies on the x-axis from 10 to 20x. And either way you look at it, we'll use the left side just for an example. What you see is that ACE certainly is providing a higher level of gene finishing within the medically interpretable genome. And so if you're looking at the solid lines, which are looking at it at an exon level rather than a coding base or a single base level, what you're seeing here is that ACE is finishing approximately 90% of the genes in the medically interpretable genome, much higher compared to all of the other providers. And so you can see whole genome sequencing at the bottom, right around 10% of genes finished in the medically interpretable genome. And the range for whole exome sequencing, the remainder of these 4 platforms, is really anywhere between about 30% to around 60–65% with other platforms. So we also looked at, as I've mentioned before, GC content or regions of the genome that are really difficult to capture with some other exome sequencing platforms, and then even whole genome sequencing. And so what you see here is the percent of finishing from 0 to 100% on the y-axis as the GC content increases on the x-axis. And so the important concept here is, again, looking at ACE and green, even as the GC content increases, the level of finishing is able to remain relatively steady. Whereas if you look around 50% GC content, you see other platforms, and specifically whole genome sequencing, really start to drop in their ability to really finish these genes. And again, these aren't specifically targeted by these platforms as they are with ACE. And so that really shows when we're looking specifically at this concept of gene finishing. So this slide is, of course, relatively busy, but what I want to do is sort of is break down the discussion in a few points and really talk about a few different aspects of looking at coverage of specifically these 56 genes that are recommended by the ACMG for reporting secondary findings with next-gen sequencing testing, such as exome and genome testing. And so, as I'm sure you have imagined, these ACMG 56 genes are included in the medically interpretable genome in those 5,419 gene set. There's a number of papers out there that really talk a lot about some of the inaccuracies in next-gen sequencing with regard to detecting variants in these genes. And so we really wanted to take a specific approach to look at these genes and also variants in these genes. And so the first part to point out is on the bottom is looking at actual gene finishing. Again, having 100% of the coding bases covered at a minimum of 20x, and how well all of the different platforms, including ACE, did when looking at it from that sort of standard. And what you see is that there's not any of the 6 platforms, including ACE, that can completely finish all 56 of those genes with their exome or genome sequencing approach. However, again, if we're looking at 100% of bases covered at 20x or greater, ACE does finish most of those genes, and we're finishing 51 out of 56 of those genes. If we look at the other 4 whole exome sequencing providers, there's a range anywhere between those platforms finishing 12 genes all the way to finishing around 39 genes. And then when you look at whole genome sequencing, what you're seeing is of those 56 genes, only 2 of those genes are actually finished according to this level of coverage here, this sort of stringent standard. So now what we'll do is talk a little bit more about this figure at the top. In this figure, we're not actually talking about whole gene finishing, but instead we're looking at disease-associated variants within these genes. And so just a little, to focus on this in a little bit more detail so you can see it a little bit better, I took a snapshot of the left-hand side of this figure so we can look at that in more detail. And so, you know, again, this is just the left-hand side looking at, I think there's around 14 genes on this zoomed-in version. But what you see here is that looking at some of the known disease-associated variants is that there's quite a bit of variation in gene coverage, and you know, as you can see, all of the platforms, you know, had inferior coverage in some areas. We looked at over 12,000 documented variants in these genes and determined what the sequencing coverage was with all of the platforms. And so you can see whole genome sequencing is represented by the gray square here, ACE by the green triangle. And then for whole exome sequencing, just to sort of keep this slide as minimal as we possibly could, we actually took the highest coverage from the 4 whole exome sequencing platforms and only plotted the one with the highest coverage. And that's represented by the purple circle here. And so what you see is, again, sampling of 14 genes with the percentage of the variants finished on the Y and the genes and the number of variants finished on the x-axis. And so all of these, again, showed pitfalls in these genes. Up to 9.6% of variants were missed with one of the whole exome sequencing platforms. Up to about 6% were missed with whole genome sequencing. And as I mentioned before, ACE also missed some of these sequence variants as well, but only 0.2% were missed with ACE. One part to really point out, just to sort of go into this in a little bit more detail, is a gene that really looks sort of bad across all of these platforms, which is MSH6. Again, you see 98% of these disease-associated variants are finished by ACE and one of the whole exome sequencing platforms, and 95% by whole genome sequencing. So we'll talk about that one in a little bit more detail, just because I think it's a good example to really illustrate coverage across platforms. So just for a little bit more reference, what you're looking at here is Personalis's blue-green coverage plots, and we use these quite often, you know, on our website and in our customer-facing brochures, because they really highlight the level of coverage with standard exome versus what we provide with ACE. And so what you're seeing here in blue is the level of coverage, or the depth of coverage on the y-axis, with a standard exome sequencing kit. In this case, this is Agilent's Clinical Research Exome. The top plot is the coding exons concatenated together. The bottom plot is the full content of this gene, so coding and non-coding regions here. And again, the blue is what a standard exome sequencing provider might cover. The green is the supplemental coverage provided by ACE. And again, this dotted line is going to that sort of clinical grade level of coverage, looking at 20x here. And so what you see when you look at the top plot is that, you know, looking at this 20x level of coverage, looking at the code regions, there's pretty good coverage utilizing a standard exome sequencing capture kit, and there's not much supplementation that we really need to do at Personalis in these regions, other than possibly adding some supplementation here, because you've got a little bit of a dip in coverage that on average may not be covered at 20x. But where the supplementation really comes in with ACE is in the non-coding regions, where we are supplementing areas here because there's known variants in these regions. But the part to really point out is that even using our ACE supplement, we're still not obtaining a level of coverage for this particular region at 20x. And I think that really illustrates how, you know, with this particular gene, this is just a very difficult to sequence gene using your standard kits out there, but also with our supplementation, we still aren't going to get 100% sort of confidence in these variant calls in this region, and that might explain some of the variation that we saw in the slide before, particularly with the MSH6 gene. And so to summarize the article, you know, this is really the first comparison looking at augmented sequencing approach compared to other commercial exomes and whole genome sequencing, but really evaluating their ability to reach a clinical level of coverage looking at these key medically interpretable genes. This analysis, of course, you know, based on what we've shown, really highlights some key gains for the ACE augmented exome approach and finishing compared to these other platforms. This is really important both for clinical applications, but it also has utility in cancer research and tumor profiling, and these are all areas that are supported by ACE technology. So now that we've, you know, gone over a background of Personalis and sort of what our ACE platform is, as well as talked about some of the highlights from the recently published Genome Medicine article, what I want to do now is switch gears a little bit and talk about how these certain augmentations with ACE and our ACE Clinical Exome platform really highlight some clinically relevant performance measures and also some case studies I think this is, a really common question or area that comes up in the clinical setting when we're meeting with people out in the field is, it's great that you're augmenting all of these important regions, but do you have any examples of when this really matters or where it makes a difference? And so that's what we'll talk about next. And so our ACE enhancements really play out in a couple of different ways in the clinical setting. And so that's what you see here represented by these two different pie charts. The one on the left is really breaking down the variants that we're reporting with our clinical exome sequencing analysis. And so what you see is, not surprisingly, approximately 70% of the variants that we're reporting clinically are variants that are picked up by a base exome. And so something like Agilent SureSelect, Clinical Research Exome, or Nimblegen or Nextera, these are variants that could potentially be identified by one or all of those exome capture kits. However, close to 30% of variants that we're reporting with ACE are actually variants that wouldn't have been picked up by a base exome, either because the larger piece of the pie here, around 23%, are poorly covered by other exome sequencing providers and need additional supplementation, such as areas where there's high GC content, or aren't covered at all in exome sequencing design. And this could be, say, an intronic variant that we're specifically targeting with our design because we know there's a medically relevant variant in that region, for example. Diagnostic yield is also an area where we, of course, have seen vast enhancements or improvements in the clinical setting. Our diagnostic yield at Personalis with our ACE clinical exome, is really right around 50% and has been steady in the couple of years that we've had exome sequencing available. Of course, exome sequencing, the sequencing side of ACE really is causing a lot of the sort of advancements or higher diagnostic yield than what you might see in other clinical laboratories. But as we pointed out with ACE, our ACE improvements aren't just on the sequencing side. We've also made improvements at every step of the workflow with next-gen sequencing, and so those areas have also resulted in this higher diagnostic yield. And so we really think of ACE as being more of an all-in-one solution when compared to other exome sequencing technologies. And so what you see here is that with other offerings, you have broad coverage of an exome, which is sort of standard across the board exome sequencing. You also have that level of gene finishing with single gene or panel tests that's going to have much more complete or comprehensive coverage when compared to broad exome coverage. And then a whole other part of the puzzle that we haven't talked about much is detection of large structural variants, which is typically not available through your standard single gene panel technologies or broad exome sequencing technologies. And though we haven't really talked about it much during this presentation since we're focusing more on the Genome Medicine publication, our ACE clinical exome really combines all three of these. We have the broad coverage, we have the finished gene coverage that we've talked about, but we also do have the ability to detect large structural variation genome-wide in our design of ACE. And so by combining all three of these into one test, it ends up being a much more comprehensive test and more cost-effective and time-efficient in the workup of patients in clinic. So again, what does all this mean in the real-world setting or in the clinical setting? We'll talk about a couple of case studies next to really illustrate the points that we've mentioned up to this point. So the first case presentation is a pair of identical twin brothers who had been diagnosed with infantile onset rod cone demonstrated retinitis pigmentosa, or RP. They also had a diagnosis of high myopia, and there was no additional family history. As with most individuals who come for exome sequencing, no different with the tests we do at Personalis, there's certainly been some prior testing that was completed before we tested these kids. One of the tests was a negative 13-gene autosomal recessive retinitis pigmentosa panel, And then the other test that they had done prior to us testing them was actually an exome sequencing test. Both, again, were negative, and so we didn't have a diagnosis prior to exome sequencing, of course. So as most of you probably know, retinitis pigmentosa is an extremely genetically heterogeneous condition. There's dozens of genes that have been implicated with retinitis pigmentosa. One of the more common ones is a gene called RPGR. RPGR is associated with around, I would say, 20% of all cases of retinitis pigmentosa, but a very large portion of X-linked retinitis pigmentosa, so around 80%. Again, going back to those blue-green plots that we were showing earlier for MSH6, this is an example of the coverage for RPGR. And so this is looking at standard exome sequencing platforms and what that coverage might look like on a standard or off-the-shelf exome sequencing kit through any other clinical lab. And what you see here, again, using that 20x level of coverage, is you see a very, very wide, obvious gap in a specific exon called ORF15. And this is an exon that's really, really well known for having poor coverage using next-gen sequencing technology due to its really, really high GC content. And so the downside of this is that approximately 50% of the variants that are in RPGR happen to fall in this region here. So any next-gen sequencing panel or exome sequencing analysis really has a very, very low chance of identifying a variant in this region. This is an example of, again, showing the augmentation or the additional supplement that's provided by ACE. And so, again, what we really focus on with the supplementation is going into these regions like the one that you see here where there's really poor coverage that's falling under that 20x threshold. And so we're able to fill in these gaps where other providers are falling short. That was really important in this particular case because in this family, that's where this variant resided. This was the variant that was found to be associated with X-linked retinitis pigmentosa in these two twins. And this really explains why, well, first, they tested with an autosomal recessive panel initially, so this gene wouldn't have even been on that panel. But then secondly, with them having negative exome sequencing, this would explain why that test would have been negative because it wouldn't have been covering this particular region here. The second case to share with you is a case of a female who had been diagnosed with mild distal motor and sensory neuropathy. Her father was more severely affected than she, and prior to having exome sequencing through Personalis, we weren't aware of any prior testing that had been completed. We found a pathogenic, previously described variant in a gene called GJB1. This is a gene that's well known to be associated with Charcot-Marie-Tooth disease. It's causative of CMT X type 1, so an X-linked form. And so this really, of course, being a previously described pathogenic variant, fit well in line with the phenotype and with the family history of this individual having milder symptoms than her more severely affected father. So this was, really a slam dunk diagnosis in terms of it really fitting the phenotype well. The interesting part on the sequencing side is how we found this variant or where we found this variant. So again, showing these coverage plots again, this is looking at sort of the full content of the GJB1 gene. And, again, if you're using that sort of 20x level of coverage, how we're supplementing this gene quite well. But most importantly, the area where we found this variant was in the promoter region. And so this was a specific region that our ACE technology targets because we know there are previously described variants in this region, such as the one we found in this case. When we went and looked back at this particular variant, we realized that a number of next-gen sequencing panels aren't targeting this in their design, even though they tend to be doing full sequencing. And of course, standard exome sequencing isn't targeting this region because it's part of the non-exonic or non-coding region. And so this was again, a specific area that we targeted in our design, and we were able to find a pathogenic variant in this particular individual. So to conclude, what we've talked about today in terms of our study and, of course, our case examples is that, we really feel that there's a number of important conclusions that are really apparent, again, from our own experience at Personalis, but also the recent findings in the Genome Medicine publication. First, we feel like clinicians really should consider analytical performance when making clinical assessments or choosing laboratories in which to perform testing. Even a few misvariants can lead to reporting false negatives. Our augmented approach at Personalis is, of course, it's an innovative strategy that provides a level of coverage that's not achievable with other platforms, and so it helps to address concerns regarding the lack of sensitivity in some of these clinically or medically important regions. And of course, in the clinical setting, an augmented approach to exome sequencing really focusing on medical relevant areas can be cost effective and can lead to diagnoses earlier and hopefully with fewer tests than current genetic workups. And so at this point, this concludes the presentation portion of this webinar. What we'll move to next is answering any questions that anyone might have asked throughout the presentation or having further discussion. I'd like to point out here, if you have further questions that we aren't able to address during today's presentation or have questions at a later time, you're welcome to send me an email with questions or comments. Please check out our website, and since this is an audience geared towards those of you attending AMP, please come visit us at our booth. I'll be there along with a few other members of our team at Personalis at booth 1711.
All right, ladies and gentlemen, so we've now come to the question and answer session. I shall read questions from the audience for all to hear before Heather responds. And again, as Heather said, if we're unable to respond to any of your questions during this time, please feel free to reach out to us by email, and we will do our best to respond to each in a timely manner. Okay, Heather, our first question is, do you cover all of the ACMG genes at 20x or greater?
Thanks, Felicia. So we talked about this a tiny bit as part of some of the slides covering the ACMG genes, and we end up finishing approximately 51 out of the 56 genes at 20x or greater. I don't have a list of each one that we don't finish at 20x, but I know just from looking at these stats that I know MEN1 is a gene that's particularly difficult, across all testing platforms, and of course we talked about MSH6 as well.
Okay, Heather. So the next question we have is, you mentioned that ACE includes improvements in other areas aside from sequencing. What else has Personalis done that is different or improved?
Sure, yeah, and I mean the majority of this presentation is obviously focusing on sequencing because that was really what we focused on for the Genome Medicine article. But ACE and next-gen sequencing, we really think of it as having those four components: the sequencing, alignment and variant calling, annotation, and of course interpretation. And so we have taken each step of that process and made improvements. One example from an alignment standpoint is we have our own Personalis major allele reference because we've realized that there are a number of minor alleles that are part of the current reference sequence, and so we've made over 1 million substitutions in the reference sequence to sort of more accurately represent the major allele, which would improve variant calling. We incorporate a number of different databases in our annotations. We actually integrate and update over 40 public databases, and we also have our own Personalis variant disease database, which is an effort internally to manually curate thousands and thousands of papers that are sort of hot off the press, and so that gives us the ability to have really the latest and greatest information from our databases. And then, of course, for clinical exome sequencing, we take a lot of pride in our approach to the interpretation part at the end. by having a really, really stringent manually reviewing process where we're ranking variants based on their overlap with phenotype, which helps to really streamline our approach and what we're doing to help increase diagnoses. So again, just a snapshot of what we do differently at each step along the way.
Okay, so the next question we have is, what about the coverage of PMS2 and CHEK2 genes?
I don't have them all remembered off the top of my head, of course. Whoever asked the specific question about those genes, if you want to send me an email to the address that you see here, I'm happy to provide coverage for those specific genes. We definitely have data available that shows coverage with our ACE platform compared to other standard exome sequencing platforms, and I'm more than happy to share that with you.
Okay, thank you, Heather. And the last question that we have time for today is, what do you guys do to obtain coverage in areas with high GC content?
It's a good question. I think, obviously the areas of GC content are of course the areas I think that are really quite difficult to sequence using standard library preparation and sequencing technologies. And what we do at Personalis is, of course, a proprietary approach with very specific library preparation that's performed prior to sequencing. And so the sequencing itself is all done at one time by pulling DNA from our standard analysis, but also from these additional library preparations.
Thank you, Heather. So we have now come to the end of our time today. Thank you all very much for joining us.